Biological weapons, a chemical perspective. Biological weapons, a chemical perspective. In the realm of warfare, the evolution of weaponry has seen various stages of transformation, from primitive tools to sophisticated instruments of destruction. One such avenue that has garnered both fascination and apprehension is the development and deployment of biological weapons. These weapons, which exploit the deadly potential of living organisms or their byproducts, have raised ethical, moral, and global security concerns. To comprehend the intricate nature of biological weapons, it is crucial to delve into their chemical underpinnings and the broader implications they carry. Understanding biological weapons. Biological weapons encompass a diverse array of agents, ranging from bacteria and viruses to toxins produced by living organisms. Unlike conventional weapons, which rely on the physical force of explosions or projectiles, biological weapons exploit the biological processes of living organisms to inflict harm. This paradigm shift from mechanical to biological action underscores the chemical nature of these weapons. The chemistry of biological weapons revolves around the intricate interactions between biomolecules, cellular structures, and biochemical pathways. Toxins, for instance, are chemical compounds produced by microorganisms that can cause severe harm or death in even minute quantities. These compounds can disrupt crucial cellular functions by inhibiting enzymes or altering cell membrane permeability. By tampering with the delicate equilibrium of biochemical reactions, toxins effectively dismantle the machinery of life. Biological agents and their chemical interactions. 1. Bacteria and viruses. Bacterial and viral agents are often used in biological weapons due to their contagious nature and the potential to cause widespread devastation. These agents manipulate the chemical processes within host organisms, hijacking cellular machinery to replicate and propagate. Viruses, composed of genetic material encapsulated in a protein coat, infiltrate host cells and integrate their genetic material into the host's DNA. This process alters the host protein synthesis, commandeering the cell to produce viral components. Bacteria, on the other hand, can secrete toxins that disrupt normal physiological functions. The chemical interactions between the pathogenic agent and the host are pivotal in determining the severity and outcome of an infection. 2. Toxins. Toxins produced by microorganisms are potent chemical compounds that interfere with cellular processes. Botulinum toxin, for instance, prevents nerve cells from transmitting signals, leading to paralysis. Ricin, derived from castor beans, disrupts protein synthesis in cells, causing cell death. The chemical structure of these toxins allows them to specifically target particular cell types or biochemical pathways, making them effective tools for incapacitation or lethality. Weaponization and delivery. Weaponizing biological agents involves refining and optimizing their delivery mechanisms. The chemical process of weaponization encompasses stabilizing the agents to ensure viability, enhancing their virulence, and devising efficient methods of dispersal. Encapsulating biological agents in particles that protect them from environmental degradation is a crucial step in maintaining their potency. The chemistry behind these protective coatings ensures that the agents remain viable until they reach their intended target. Delivery mechanisms for biological weapons vary widely and often involve the use of aerosols, contaminated surfaces, or contaminated food and water supplies. Aerosol dispersal relies on the principles of particle science, where the size and characteristics of particles determine their behavior in the air. By engineering particles of specific sizes, the dispersal efficiency and reach of the biological agent can be controlled. The chemical properties of these particles also play a role in their adhesion to surfaces, affecting the persistence and spread of contamination. Chemical detection and defense. The specter of biological weapons has spurred intensive research into chemical detection and defense strategies. Detecting the presence of biological agents in a timely and accurate manner is crucial for mitigating their impact. Biosensors that exploit the specific interactions between biological agents and certain biomolecules have been developed. These sensors can detect minute quantities of agents through changes in electrical conductivity, fluorescence, or other chemical properties. In terms of defense, understanding the chemical interactions between host organisms and biological agents is paramount. Developing vaccines and treatments that target these interactions can provide effective countermeasures. Vaccines elicit an immune response that prepares the body to recognize and neutralize specific pathogens, effectively providing immunity. Antiviral and antibiotic treatments are designed to disrupt the chemical processes essential for pathogen survival and replication. The development of these defenses hinges on an in-depth understanding of the biochemical intricacies of pathogen-host interactions. Ethical and global implications. The chemical perspective on biological weapons inevitably leads to a consideration of their ethical and global implications. The use of biological weapons raises questions about the moral boundaries of warfare and the potential for indiscriminate harm. Unlike conventional weapons, biological agents can spread beyond intended targets, affecting civilians and the environment. This uncontrolled dissemination accentuates the necessity for strict regulation and international agreements to prevent the development and use of such weapons. The enforcement of these conventions requires cooperation among nations, robust monitoring mechanisms, and effective sanctions to deter noncompliance. The chemical aspect of biological weapons plays a pivotal role in devising verification mechanisms and monitoring protocols to ensure compliance with these agreements. Conclusion. Biological weapons, with their profound implications for security and ethical considerations, are an embodiment of the convergence between chemistry and warfare. The synthesis of these elements is crucial in navigating the delicate balance between scientific progress and the preservation of human well-being. Emerging challenges and future prospects. While significant strides have been made in understanding and addressing the chemical aspects of biological weapons, new challenges and prospects continue to emerge on the horizon. The rapid advancements in biotechnology, genetic engineering, and synthetic biology have the potential to reshape the landscape of biological warfare. These technologies offer unprecedented capabilities to modify existing pathogens or create novel ones, blurring the lines between naturally occurring agents and engineered threats. The chemical perspective on biological weapons must adapt to these evolving challenges. This involves staying abreast of cutting-edge research in the life sciences and continually updating detection and defense strategies.